अभी हम बात करेंगे ग्रोथ कर्व के हवाले से। बायोटेक्नोलॉजिकल लैब में माइक्रो ऑर्गेनिज्म के साथ काम किया जाता है। मोस्ट ऑफ द टाइम वी यूज बैक्टीरिया और अदर माइक्रो ऑर्गेनिजम्स, एंड टू start working with any microorganism, we need to have some idea, some knowledge about their growth pattern. Because knowledge about their growth pattern can make it sure to get reproducible transformation, reproducible efficiency, plasmid or protein yield. If the microorganism which is under use don't have good growth potential, then it is useless to use them as the host cell for transformation or for any other procedure. When we talk about the bacterial life cycle or bacterial growth curve, it has different phases. आपको पता होगा, जैसा कि आप जानते हैं, लैग फेज, लॉग फेज, स्टेशनरी फेज एंड देन डिक्लाइन और डेथ फेज। ये वो 4 फेजेस हैं जिसमें बैक्टीरिया अपनी ग्रोथ कंप्लीट करता है। लैग फेज को देखा जाए तो ये वो स्टेज है जब बैक्टीरिया अभी डिवीजन की तरफ नहीं जाता। एनी बैक्टीरियल सेल इज गेटिंग मैच्योर, एंड वंस द मैच्योरिटी इज अचीव्ड, then there comes an exponential increase, which is called as log phase. As the time passes, nutrients are consumed, there are the factors which are required by the bacterial cell for division, but they are not available. Then there comes a stationary phase when growth limiting factors overcome and the bacterial division stops. This is called as stationary phase. And after some time period, due to some physiological factors, environmental temperature, surrounding medium composition, the bacterial cell starts dying. And this goes towards the death phase or decline. Whenever we want to study about the bacterial growth potential, we need to start with the uniform balanced growth. And we get the uniform balanced growth idea from the log phase of any bacterial growth curve. Log phase will give us an idea what is the growth potential of a particular bacterium. Bacterial growth curve or any microorganism's growth curve depends upon the media composition. हम जब बैक्टीरिया को ग्रो करते हैं, हमारे पास मीडिया की चॉइस होती है. Depending upon the species, depending upon the strain of the bacteria, we modify the medium. But broadly speaking, we use either minimal medium or we use rich medium. Minimal medium is not supplemented with essential nutrients. Complex medium is supplemented with essential nutrients. If you culture bacteria in minimal medium, its doubling time is increased. This is because of the fact the bacteria has to synthesize all the nutrients by the metabolism. As you can see in this table, in minimal medium, bacteria needs 50 to 60 minutes for doubling. In complex medium, it can divide within 20 to 30 minutes. Same is with the yeast, 200 minutes in minimal medium, 100 minutes in rich medium. और जैसा कि मैंने पहले बताया कि मीडिया चॉइस इज इम्पॉर्टेंट, कीपिंग इन व्यू what type of bacterium or microorganism you are using, whether it needs a minimal medium or it needs a complex medium. And then the minimal medium is going to take more time then the bacteria is going to take more time in minimal medium for its division. Keeping in view the type of media, the onset of log phase is different. हम ये नहीं कह सकते कि एक बैक्टीरिया मिनिमल मीडियम में अगर 5 मिनट के बाद लॉग फेज अचीव कर रहा है तो कॉम्प्लेक्स मीडियम में भी वो 5 मिनट के बाद ही लॉग फेज में जाएगा. कॉम्प्लेक्स मीडिया में जो लॉग फेज कैन बी अचीव्ड अर्लियर एज कंपेयर टू ग्रोइंग इन द मिनिमल मीडियम. Besides the nutrients, there is requirement of oxygen supply. And for that purpose, for homogeneous distribution of oxygen, vigorous shaking is required of the culture flask or of the test tubes. Because if you don't do the vigorous shaking, then from a particular part of the culture media, there will be oxygen deficiency. And that oxygen deficiency may interfere with the growth pattern of the bacteria or any other microorganism.